Samuels. I work in the botany department and I'm the academic director of the UBC BioImaging lab, which you're going to hear more about, bioimaging facility that you're going to hear more about today. I study how plants make cellulose and lignin, and it's really nice to have this interdisciplinary group for learning and discussion. So I must say that this seminar or this session will be recorded, and I would like to encourage all attendees to join the mailing list by the QR code displayed on the slides. And now it's a pleasure to give a brief introduction to my colleague, Dr. Miki Fujita. Dr. Fujita did her undergraduate degree in Tokyo at the International Christian University, and then went to Australia to start a PhD and ended up coming to Vancouver with her PI, Jeffrey Wastenese, and finished her PhD here at UBC studying how plants make cellulose, and more specifically how they organize that cellulose to determine the three-dimensional shape of plants. And so since 2018, Dr. Fujita has been the manager of the UBC BioImaging facility. So she's the point person when new people come in with projects. She helps them find the right resources and helps to manage the projects and advise people on the best infrastructure to use. So without further ado, I'll turn it to Miki.
Thank you, Lacey, and thank you for coming to this seminar today. And I'm going to talk about our facility and this slide. So UBC BioImaging facility is located in the Biological Sciences building. It's next to bookstore. And this photo is taken from the water fountain at the cross of Main Mall and University Boulevard. And if you take this door to North Wing and go into downstairs, and you can find us at the basement. And we are open to everyone, and we provide access to optical microscopes, to electron microscope, and you can do sample preparation, imaging, and image analysis in our facility. And we also provide training and service. And we have the website set up, bioimaging.ubc.ca, and you can find most of the information I'm going to talk about today. So here is our website, and I would like to introduce our team. So from left to right, we have Azuki, undergraduate research assistant, and Reichi as academic director, Yoon Kyung Lee, who is optical microscopy staff, and Lari helps us with fifth sem project, and Derek Hong is staff for SEM and TM, and Jesus is our TM staff, and myself, Arash is helping us with cryo TM project, and Rafi comes once or twice a week. If you're lucky, you can meet him. And so what we offer, we have optical microscopy. Currently, we have 4 optical microscopes. You can do light fluorescence imaging, confocal imaging, lifestyle imaging. And also we have additional advanced fluorescence techniques like multiphoton imaging, second harmonic generation, and fluorescence lifetime microscopy imaging. The optical microscopy, the theoretical resolution limit is 200 nanometer. And one of our optical microscope can do a super resolution imaging, and this resolution is XY resolution limit. If you can go up to 120 nanometer. But if you have samples that are very small nanometer level, or if you're gonna get high resolution image, then you may want to do electron microscopy. So currently we have 2 SEMs scanning electron microscopes. Resolution limit is 1 nanometer. And we can do regular conventional SEM imaging, also elemental analysis using energy dispersive X-ray spectroscopy. And also we can do cryo-SEM, so we can look at frozen samples. And it has also capability of doing focus ion beam SEM and cryo-FIB SEM. And we also have 2 TMs that we can do conventional TM, high resolution TM, cryo TM, also electron tomography, and correlative light and electron microscopy. The bolded capabilities are the one that are unique to our facility among all the UBC facilities. And we have sample preparation equipment, photos and dryer for SEM sample preparation. Also, we have sectioning equipment and also cryo electron microscopy processing equipment. So I'm gonna go over the capabilities we have. So I would like to also talk about our users bringing a variety of sample types. And there are material samples and biological samples. And I wanna ask you, how many of you working on material samples? And how about biological samples? Okay, some more material samples. So the example of material samples users bringing the aerogels, hydrogels, polymer films, plastics, graphene oxide, and silicon nanoparticles, quantum dots. And also we have a lot of users coming from bioproduct institutes, and those users bringing cellulose or lignins extracted from plant tissues, and also some of the materials are extracted from animal tissues. So those ones are nanocrystals, fibers, nanoparticles. And biological samples, we have a variety of biological samples, users bringing virus, bacteria, and cell cultures, animal and plant tissues, and insects, and also sometimes cellular components extracted from cells. So I'd like to start with the optical microscopes we have at the BIF. The first one is sterile microscope. It is also used as a dissecting microscope. You can bring the bulk sample. And nice thing about our microscope is that it has a good high resolution digital camera that can do monochrome and color image acquisition. So you can do all the imaging with those cameras and capture all the samples. And also we have the light source coming from the top of the lens and also bottom of the lens. And also we have side lights, lights coming from the side. So you can adjust the light condition before taking images. And then moving on to second one, light fluorescence microscope. We have Olympus PX53. This is a regular compound microscope. It has camera on top. It's also high resolution monochrome color camera and binocular here. And then here is the objective lens and stage here. We have a variety of objective lenses, and you can do bright field, dark field imaging. Also, if you have samples that are transparent and clear samples, we have the tools to increase the contrast using phase contrast or differential interference contrast imaging. And also this one can do fluorescence imaging. For your samples, you have to mount them in a glass slide and cover slips because there's not much distance between objective lens and stage. If you want to count cells or your molecules or want to see that find out the density, we have the slide with the grid. It's called hemocytometer. So you can count your samples in a grid and then measure and calculate the density of your samples. So this is an example of some material samples. Here on the right is the pickering emulsions made of cellulose nanofibrils and phase change materials. You can see the sizes and density. We can also use the hemocytometer to get the density information. And those two images are showing the same samples. It's enzyme microcrystals, but the middle edge image is showing a regular bright field image. And then this corner image is showing the phase contrast image. So it's increasing the contrast using phase lens. And what it does is it creates halo effect around the sample. And kind of it's good to look at sample edges. So sometimes we use the phase contrast imaging for clear transparent sample. And this microscope has capability of fluorescence microscope imaging. So we use the mercury arc lamp or LED lamp as an illumination source, and illumination light source coming from the back of the microscope. And in the middle of the microscope, it has a we call it filter cube that has excitation filter, emission filter, and dichroic mirror. What it does is to select a certain wavelength. So in this case, excitation filter is selecting blue light, and blue light reflected by dichroic mirror traveling to objective lens and illuminate the sample. The flow force in the sample absorb photon and get excited. And before they go back to ground state, they release energy as a fluorescence light. So usually if we use short wavelength light, then emit the light long wavelength light. So in this case, green light reflected back to objective lens and passing through dichroic mirror, and emission filter narrows down the wavelengths to get detected by detectors of binocular. So this fluorescence technique is used for clear samples like oil droplets. Oil droplets is very challenging to see it under bright field or phase contrast imaging tools. Then we can stain them with fluorescent dyes. So as in a black background, we can see them clearly, and we can do analysis of size or density measurement. And this is another example of plant samples. This is the differential interference contrast image. Without labeling, we can see kind of a topological effect of the samples. Here is the image taken with color camera, and plant samples have autofluorescence. So it is showing the cell and then chloroplast. The bottom image is the plant root samples stained with different dyes. And this image actually was it's a stitched image of high resolution image. So there's a software that can do automated multi image acquisition, and then can stitch together to make a large image. I'm gonna move to confocal microscopy. We have Leica Stellaris 8. It was installed 4 months ago. It uses continuous laser to excite the flow pore, and there's a detector pinhole to eliminate out of focus light. And this confocal system is useful for optical slicing and 3D imaging. So if you have fluorescence samples and want to see from top to bottom of your samples, what we can do is optical slicing. So this is the example of 3D reconstruction of leaf tissue. So here is the color indicator. If it's red, it's really close to the surface of the leaf, and as it goes deep to the tissue, and then color changes to blue. So it's based on the 2D image of optical slices, we can reconstruct them and then color code it. And this is the left one, right here. Right one is the fungus infecting the plant tissue. So red means it's really close to the surface and goes to deeper. And I'm not sure if you can see, but as a background, you can see the leaf surface. And with the software, we can make movie. So it's showing again fungus hyphae infecting the plant leaf surface as red. And we can go deeper and deeper and deeper. It's about 120 microns deep from the surface. And from these, we can make also 3D representation movie of the fungi and how they enter into the leaf tissue. So we can do make I think it's for presentation. It's very great tools. This Leica Stellaris 8 microscope has another capabilities called multi-photon microscopy. So this microscope is equipped with pulsed tunable lasers, and pulsed lasers are used to excite fluorophore with 2 or more photons. And usually we use infrared light wavelengths to illuminate the sample. And those longer wavelengths light, they can penetrate tissues deeper, samples deeper. So it is used for deep imaging up to 750 microns. Previously, I showed you the confocal X3D reconstruction. It was up to 250 microns. So multi-photon can go deeper. And with pulsed laser, we can also do second harmonic generation imaging. We call it SHG. Pulsed tunable laser is used for that. What is happening is that 2 photons are converted to 1 photon by sample. And the incoming wavelength frequency gets doubled after interacting with sample. So if we are using, let's say, 900 nanometer incoming wavelengths to illuminate the sample, then after interacting with samples, it gives 450 nanometer wavelengths. And this is not fluorescence phenomena, it's just light scattering and useful tool for highly anisotropic type alignment of repeating structures. So it doesn't happen to all samples, just the samples that have highly ordered and tight alignment structures. And the example of this would be collagen fibers and muscle tissues, neurons. Also, if the samples have crystalline structure like crystalline cellulose and starch granules, we can get this. So this is an example of SHG and multi-photon imaging. So Robin Birch from Ritzy One Lab has bone has a lot of collagen fibers. So that with SHG imaging, she was able to see the bone structure. And using the same pulsed laser illumination light, she was able to do multi-photon imaging. Here the green shows blood cells. Blood cell had autofluorescence. And beauty about this technique is that we use the same excitation pulsed laser around 810 nanometer wavelengths. Then we set up 2 tunable detectors, one to detect second harmonic generation and another detector for multi-photon. And the last capability of this Leica Stellaris 8 microscope is fluorescence lifetime imaging microscopy called FLIM. So this technique, we also use pulsed laser, and we measure the time between laser pulse and fluorescence photon is detected. It's called arrival time. And this photon arrival time is recorded over time, and it creates histogram of photon arrival time, this photon. And it scans across the samples that this one is the image of samples, and each pixel will show those photon arrival time histogram. And based on that, the system creates color-coded fluorescence lifetime image. So if the fluorescence lifetime is short in a certain area, it shows red. And if it has longer fluorescence lifetime, then it shows green bluish color. And if there's no fluorescence photon is counted, then it will show. Oh, here it's white, but it's going to be black. And what is fluorescence lifetime? So fluorescence lifetime is unique to each fluorophore. And also it's, but it can be influenced by surrounding environment like temperature, pH, ion condition, and molecular interactions. But it doesn't affected by fluorescence concentration or what kind of illumination light wavelength was used or if it's photobleached or not. And it has been used for many biological samples in our facility. This is the example of marine organisms. The DNA was stained with fluorescence dyes. So this is fluorescence image based on the intensity. So it shows all the localization of DNA containing organelle. But if we do film fluorescence lifetime image, we can see differences in some compartments. So red shows short fluorescence lifetime, and blue shows longer fluorescence lifetime. Kind of indicating that there are different compartments in organisms, and then the environment is different. Another useful tool using film is we can separate fluorescence signals from autofluorescence background. So a lot of biological samples have intrinsic fluorescence, and it has different fluorescence lifetime. And here the example that Coke pot, biological samples had a lot of autofluorescence, and based on the lifetime, the researcher was able to separate fluorescence signals from autofluorescence background. The last computer microscope we have is Evidence Spin SL. It has a variety of objective lenses. We have dual camera systems and solar spinning disk system, and it is used for high-speed, multicolor lifestyle imaging. Also, it can do super resolution imaging, the XY resolution 120 nanometer. And this is an example of plant cells expressing the fluorescently labeled protein. And here is the multicolor, two-color imaging of also fluorescently labeled proteins in the plant cell. Any questions so far about the optical microscope? Yeah, I'm going to move to sample preparation equipment. We have sectioning equipment, vibratome, ultramicrotome, dryer and filters for SEM sample preparation. And we also have cryo sample preparation equipment for sample preparation and also cryo SEM, cryo TEM, and also cryo sectioning with the ultramicrotome. So I'm going to start with sectioning tools. We have Leica BT1000S vibratome. Vibratome uses razor blade vibrates high frequency, and then it cuts samples embedded in agarose. So here is the samples, and then there's a sample chamber we filled with water, and as razor blade advances close to the samples, and we can cut the cross section. And the samples have to be soft enough, like same firmness as agarose gels. And by vibratome sectioning, we can make 50 to 500 micron thick sections. And after sections are made, it tends to float in a water bath. So we use the brush to collect them and then mount them in a glass slide. And you can stain them, and you can also do the optical microscopy. So this is an example of plant stem cross sections. So the cross section was made but with vibratome and then stained with Hoyden blue, and then was imaged with Olympus BX53 microscope. Another sectioning tool is ultramicrotome. We have Leica UltraCut 7. So here is the specimen block, and here we have glass knife, this photo. And we can cut both soft and hard samples, and we use a glass knife or diamond knife. The samples, if the samples are wet, we have to dehydrate them because we want to embed them in a plastic resin block, and plastic resin doesn't polymerise if there's water content. So have to be dry before, no water before embedding the plastic resin. And for the microtome sectioning, if we are making sections for optical microscopes, we use glass knife. We can get 200 to 1000 nanometer thickness sections. And if we are doing sectioning for TEM, we use diamond knife, and we can make 40 to 200 nanometer thickness sections. So yeah, either like depends on the section thickness. They can go to optical microscope or transmission electron microscope. So I want to entertain you with the movie clip. So here is the ultramicrotome in action. So here is the sample block moving up and down, and every stroke it gets closer to the knife. Here is the diamond knife, and we call it bold. We filled with water here. And this is the top view of sample block. And here is the diamond knife. And here filled with water. And as we cut the section, so you can see sample block is moving. And you can see the ribbon of section. The plastic resin is hydrophobic and tends to float on the surface. And from there, we can collect sections for TEM or light microscope. So I want to switch to the dryer and photo. So sample requirements for STM, because STM column is highly vacuum. Samples must be dry. If there's any liquid, then it gets vaporized and contaminate the electron column. And also samples must be electrically conductive. So if your samples are wet, researchers use do the air dry or oven dry or freeze dry. But some samples are really sensitive to heat or drying process. Then we have the equipment called critical point dryer. So the samples submerged in 100% ethanol, and with using critical point dryer inside of the sample chamber, it gets replaced with liquid CO2, and increasing the chamber temperature and pressure, liquid CO2 becomes gas CO2. And we can keep the surface tension intact as possible, and critical point dryer is used for hydrogels and biological samples. And if you have beam sensitive samples, then we have to deposit thin layer of metals. And we use the sputter coater equipment, and we have choice of coating materials, different coating materials, and usually we coat them like 2 to 5 nanometer, but sometimes up to 30 nanometer of metal coating is required. It depends on the samples. And for the cryo sample preparation equipment, we have all the associated equipment to do the cryo STM and cryo FIPSEM, and also cryofixation of the hydrated samples, and cryo TM and cryo sectioning. And I want to introduce this cycle EMICE. This is a high pressure freezer we installed last year. This is used to immobilize the cellular events within milliseconds at the cryogenic temperature. The challenging thing about this equipment is this big, but then we have to have very tiny samples. It has to fit into sample holder 3 millimeter diameter. So here is the sample holder. So with 2 sample holder, we sandwich the samples and then put it in the cartridge. And then samples get frozen. And we have the system to keep frozen samples in liquid nitrogen all the time. So from there, we transfer samples from next process, or we keep them in a liquid nitrogen duo. So I'm gonna switch. Our SEMs. We currently have 2 SEMs. Hitachi S2600 is 25 years old. It's not working right now, so I'm gonna mainly talk about Zeiss Crossbeam 350. This equipment is used for high resolution SEM imaging at low accelerating voltage. Also, we can do the EDX analysis, elemental analysis. It has a large sensor 170. And also we can do cryo SEM and focus on beam SEM and cryo FIB-SEM and also TM lamellar preparation. In the coming months, we are going to install the Hitachi SU8700. This is another SEM. We can do high resolution SEM imaging at low kV. Also, it has a variable pressure mode. So we tested some non-coded samples, and we can get a very good high resolution image with using variable pressure mode. Also, it comes with the EDX capability, and also we will have STEM detector, so we can put TM samples and to see the internal structure of low atomic number materials like polymers, carbon nanotubes, and biological samples. So I'm going to talk about this Zeiss Crossbeam. So as I said, I can do high resolution imaging. This is the example of cross-section of chitin nanocrystal film. You can see, well, it's the same sample, but different magnification. And also it has a software that can do 2D tiling. So it can take multiple images and then stitch them together. And another example, Soya was looking at different carbon structures. And we also have the example of plant samples showing wax crystals and sea spider and proteus cells. And as I said, it has the capability of doing elemental analysis. So here is the example of a matched SEM image of distribution of different elements. So in this photo, you can see distribution of oxygen, carbon, and aluminum, and silica. And you can get the overlay images. And another application, as I said, we can do cryo SEM, cryo FIB, and cryo FIB-SEM, also TM lamellar preparation. This Crossbeam 350 is a dual beam focus ion beam SEM. So in the center column is the electron column, produce electrons, but we have another ion column, produce gallium ions, and we have another device called gas injection system to deposit platinum to the samples. And I have the example of FIB-SEM samples. Here we have the electrode structure made of graphite layer and manganese oxide particles embedded in activated carbon. And with these samples, deposited the platinum part on top of the samples, and then did field milling. So you can see the internal structure of the samples. Here is the platinum part, and here is the nanoparticles here. You can see. And we can do fifth SEM for the biological samples too. So here, cell is embedded in a plastic resin, and then we did the field milling and SEM images, and are getting whole cell internal structure information. And also we can rotate and then do the video clips show different structures. Based on the fifth SEM. Any questions about SEM fifth SEM? Okay, so I'm gonna move to our TEM. We have two TEMs. So for the transmission electron microscope, we use much higher accelerating voltage. Usually SEM is up to 30 kV. We use up to 200 kV of electron beam. We have two TEMs. One is for 120 kV TEM, and another one is 200 kV TEM. The conventional TEM imaging, we use around 80 to 120 kV accelerating voltage, and usually we calculate sample thickness 1 nanometer. 1 kV electron beam can penetrate 1 nanometer. So if we are making sections for conventional TEM, we usually make 70 nanometer sections, 70 to 100 nanometer sections thickness. And if we go also, if we increase the accelerating voltage, then we get higher resolution image. So if you want to look at a small structure, nanoparticles, then we use 200 kV TEM to do high resolution imaging. And also 200 kV TEM, we have capability of doing TEM and electron tomography. And the theoretical resolution of the 200 kV TEM is 0.14 nanometer resolution. So I'm gonna talk about this conventional TEM imaging, and then move on to those specialized TEM applications. So the sample requirements for conventional TEM, again, samples must be dry, and also must be electron transparent, is ultra thin, because electron beam coming from the top and then have to transmit through the samples. And if the samples have area that electron dense area, the electron gets scattered. And here is the detector, so the electrons don't get to the detector, and then area would be black area. But if there's an electron transparent region, then we get some electrons collected by detector, and so that can be a principle of image formation in a TEM. And another requirement would be samples must be small enough to fit into TEM grids, which is 3 millimeter diameter. So if you have samples in a suspension layer in a liquid, then we can have just put a drop of your samples and let it dry, and we can put in a TEM. Or if your samples are already electron sensitive, then we have to stain them using granular acetates. It can give the electron density, then we can see samples. So this is the example of the materials samples, the TEM images. Here is branched silica particles with bacterial cellulose fibers. Here is the chitin nanocrystals extracted from shrimp shells. And here is the aerosol particles. And for the the biological samples, we have to fix them and dehydrate and then embed them in a resin and then make sections, then also stain with uranium acetate and heavy metals. And we can see all the biological samples, the very nice structures like cells and tissue. It requires a lot of sample preparation for biological samples if they are tissues and cells. The TM, if your samples have crystalline structures, we can do imaging and also get diffraction pattern at the same time. The switching between imaging mode and diffraction pattern mode is really easy. We have to change the strength of the intermediate lenses and also take out one objective aperture and then put different aperture, and we can get, so this is the example of copper cubic particles. Image shows here and then diffraction pattern. So moving on to specialized applications, our 200 kV TM can do cryo TM. So samples will get frozen and kept in a liquid nitrogen temperature. And we have specialized cryo TM holder. It has the liquid nitrogen duo, so samples is kept at cryogenic temperature all the time. The good example of the examples would be liposomes made of lipids, and lipid particles, if we dry them, they just don't keep their structure, like morphology gets totally distorted. So usually researchers freeze them and then look at them at liquid nitrogen temperature. So you can see nice round shape. This image, this sample is very close to the COVID vaccine. Liposomes containing. Another applications we can do is 3D electron tomography. So Alberto from Rogat Lab, he initially used the 2D TM imaging of his soot nanoparticles, but he wanted to look at them in 3D, and he decided to do electron tomography. So he can see all the connections, all the particles, how they are connecting to each other. And yeah, I have several examples. And this electron tomography technique also used for biological samples. So here is the animal tissue showing, first of all, showing the tomograms. And then identify region of interest and color coded. And from this tomography experiment, they are able to identify receptors localized at the membrane. So moving to our training, we regularly provide a microscopy training to the grad students, postdoctoral researchers, sometimes PIs, and also researchers from the industry clients. Our optical microscopy training, we set up 2 sessions of 2-hour hands-on training sessions. And SEM and TM, we have online course set up on campus. And after you finish the online course, then we offer hands-on session. We can also offer group training if you want to get training with your lab colleague. First session can be group training, and then later you're gonna get a one-on-one training. If you wanna get 5th SEM training, you have to complete SEM training first. Then we provide 20 hours of 5th SEM hands-on training. We also offer microsome sectioning training, usually at least 1 day of hands-on training. And also we offer sample preparation training for electron. the training and service fees, when you get training, we charge equipment usage fee plus tech assistance fee. And after you complete training, then you can book the equipment and our staff will be available for on-call help. And yeah, we just charge equipment usage fee. If you have, like you want to get several images for your publication, then you can submit your samples and we provide service where you drop off the samples and our staff does the sample preparation and the imaging for you. And in that case, we charge both equipment usage fee and tech assistance fee. And if you have like more than 8 samples, and we usually recommend you to get training fees because service fees get really adds up the cost, then maybe getting training will be cost efficient for your supervisor. And for you, yeah, you can carry on. So if you're interested in getting training and service from us, go to our website and you can submit the inquiry form. Then I'm gonna contact you to schedule a meeting and we can discuss what kind of microscope you can use, or you wanna get training or service, or like how much it costs for your project. Yeah, then this is my last slide. I'd like to thank all the BIF users, and our facility is supported by botany and zoology department, and botany actually taking care of our cybersecurity system, and Crossbeam 350 SEM was acquired with the CFI folder Professor Frank McLaughlin. And most of the new equipment we installed last year, this year, was acquired with BioSys CFI grant led by Tracy and also Professor Oran Rojas. And we are supported by faculties of science, applied science, medicine, forestry, land and food systems, and PPRI. And here is my email contact. If you want to discuss your project, please feel free to email me, or you can visit our website. And also thank you for Joy and Ben